guys, I'm going to talk to you today about what you need to do for your pre-lab to get ready for the lab that we're going to do on Monday. Not only are you going to have to do the pre-lab, but please come dressed in tennis shoes. You're going to need them. We are going to be exercising, and we're going to be working with glassware, so your toes need to be covered. What we're doing in the lab, and you guys will have the chance to read through all of this, and you need to read through the entire lab before you do anything, so that way you know what's going on. But we are going to be measuring how much carbon dioxide you guys are going to be producing when you do different types of activities. Obviously, if we're making more carbon dioxide, we're just going to be going through aerobic respiration, so we should be making more energy, and that's what we're going to try to measure. So what I need to see for your pre-lab is we have these 4 questions right at the top that we need to answer. We can go ahead and have you guys read through the background. This is where you're going to get some really important information to possibly help you answer these questions and some of the questions on the next page. If we flip, let me show you what other questions we need to answer. 1 through 4 on the front is going to be pre-lab, and then I need to see 1 through 8 on the second page. We need to see a hypothesis, an if-then statement. Which type of activity, either aerobic or anaerobic, is going to produce the most carbon dioxide? So give me an if-then statement here. Here we have our procedures for titration, which I'm going to talk to you guys about. And then we actually have our procedures for the experiment. As you're reading through the experiment, you need to make your data table. Now, your data table should also include your partner's information. So this needs to be on a separate sheet of paper. Down here, there's probably not enough room for you to make your data table, and this is just going to be for your practice titration for you to record that when we're in lab on Monday. After the experiment's all said and done, you will need to make a graph. We'll talk about this on Monday. I've given you some graph paper, and then we have obviously the analysis questions to answer once we have all of our data collected. Now, if you notice, because there is 15 analysis questions, I do not ask you to write a conclusion. So I understand that this seems a little bit more than what we typically would have, but there will be no conclusion to write. Okay, here's some of the tricky parts that I wanted to walk you guys through and show you what we're actually going to be doing, so that way on Monday you guys will be ready for the lab. Now, we're going to be using bromethyl blue to help us measure the amount of carbon dioxide in a solution. If we just go ahead and read through this, when carbon dioxide enters water, it makes the water more acidic. Bromethyl blue is a chemical that changes color, blue to green, if exposed to acid. In other words, bromethyl blue will change colors from blue to green when carbon dioxide is present. Using the titration technique described below, you can compare the amount of carbon dioxide produced by people at different levels of activity. Now, in this flask that I have, see if I can get you a better view of it, I have some bromethyl blue solution. We can see it is a nice dark blue color. And when we're looking at this color of the solution, it really does help if we have a white background. So when we're doing the lab, I'm going to encourage you to have a piece of white paper there so you can see the color change. Now, I'm going to go ahead, using the straw, I'm going to gently blow into the flask. Now, this is not blow as hard as you can, because all of the bromethyl blue solution is going to splash up and get into your eyes, and that's not going to be fun. So we're going to gently blow into this until the solution turns from a blue to a green color. Okay, we should be able to see the color change now. No longer have that nice dark blue, but instead we have a kind of a pale green color. So this has shown, hey, we have made the water more acidic. Carbon dioxide is going to be present, and that carbon dioxide came from me exhaling into it. Now, with this solution, let's go ahead and take a look at our titration technique, because this is what you guys are actually going to be doing in the lab. So on the second page, under the titration technique, let's just go ahead and walk through this. It says, pour 25 mils of bromethyl blue solution into a 50 ml flask. I've already done that check. Using a straw, gently blow into the solution until the color changes from blue to green. We did that as well. Step 3, it says add 1 drop of sodium hydroxide to the flask and swirl. You will see the sodium hydroxide will briefly change the solution from blue to green. Now it's really important that we are only doing 1 drop at a time. I have, see if you guys can read that, not so much, some dilute sodium hydroxide in a dropper bottle. This is what we're going to be working with on Monday. If I squeeze the top of it, a little bit's going to come up. Maybe a little bit more is going to come up. And I'm going to just add 1 drop to the flask. Now, if you look straight down, we can see it's initially starting to turn blue right where the drop is. I'm going to gently swirl my flask, and we can see that it has a blue color, but when I keep swirling, it's going to go back to green. So we saw it briefly change from green back to blue. Step 4, keep swirling the flask until the solution returns to blue. Did not return to blue, so let's go down to step 5. We will keep adding 1 drop of sodium hydroxide to the flask at a time until the solution remains blue. So we're going to count these drops as we add them. So this was 1. My second drop, I'll swirl. It turns blue briefly. I keep swirling. It turns back to green. I'll add my third drop. It turns blue. Very briefly, turns back to green. Fourth drop, blue. I keep swirling, turns back to green. Fifth drop, blue, back to green. Obviously, the more carbon dioxide we have, the more drops it's going to take to get this back to blue. So this could take a while. Let's see how long this takes us. This is number 6. Blue to green. 7. Blue back to green. 8. Blue back to green. 9. Blue to green. 10. Blue back to green. 11. Blue back to green. 12. Blue back to green. 13. Blue to green. 14. Blue to green. 15. Blue. Back to green. 16. Blue back to green. 17. Should be getting close here. 18. Slowly trying to turn back to green. Maybe the next drop is going to fully titrate it for us. 19. See if it stays blue. Not quite there yet. Still a little bit green. Let's go 1 more drop. And this will be 20. Still looks a little green. We'll go 21. Okay. 21 looks like it might do it for us. We are back to that original blue color that we started with. It then says to swirl the flask up to 30 seconds to make sure that your color change is going to be permanent. If your solution has not turned back to the original royal blue color, add one more drop of your sodium hydroxide and swirl the flask again. The more drops of sodium hydroxide needed, the greater amount of carbon dioxide is in the flask. And then we're going to record the number of drops in the data table over here as our practice. So everyone's going to get a chance to practice before you do the experiment. If we look at the procedures for the experiment, we're going to be working in groups of 2. One of your partners, either you or your partner, is going to do the aerobic portion A. The other one's going to do the anaerobic portion B. Now, I'm going to let you guys read through this. Read through it carefully. If you want to be the aerobic portion A, this means you are going to be jogging in place for 5 minutes. My anaerobic portion B, this person is going to be doing tuck jumps for 45 seconds. So between you and your partner, determine who wants to do what. You do have to do either A or B. You cannot just sit out and watch this. So make your data table. So recap of everything that I want for you guys to do for Monday. Pre-lab 1 through 4. Obviously read through all of this information. I need to see pre-lab over here 1 through 8 and your hypothesis. And then we need to read through the experiment and make your data table, including your partner's data. If you have any questions over this, please come and see me Monday morning. We need all of the lab time during class, so if you have questions, come early and ask me. Have a good weekend.